In this tutorial we'll look at basic chromosome structure. On the left is a remarkable picture, and it shows you a diagram of chromosome number 10 on the human genome. You recognise that we have 23 pairs of chromosomes, so there were two chromosomes of number 10. And after the Human Genome Project, which was finalised in the year 2000, we have identified many of the genes along each of the human chromosomes. And this shows you just the extraordinary complexity of just one of those chromosomes. 135 million base pairs long on chromosome number 10, and you can see each of the genes labelled along the length of that chromosome. So what exactly is a chromosome, and how does it relate to the molecule of DNA, which you've already studied in this course? So let's have a look. Essentially what we call a chromosome, shown down here, is just a highly folded piece of DNA wrapped around specialised proteins called histones. So let's have a look at how this might work in practice. So if we start at this end here, we can see that there are genes that are particular lengths of the DNA molecule, which you're very familiar with by now. The DNA, of course, is shaped into a double helix, and you can see that shape here. That DNA is then wrapped around proteins called histones, and this combination of DNA and proteins together is referred to as nucleosomes, and we'll have a look at that in just a moment. Then these nucleosomes are folded up tightly, so they're multifolded like this, and ultimately this highly detailed folding of the DNA around these proteins forms the structure that we call a chromosome. Let's have a look at this in a little bit more detail. We can see here another scheme here that shows you this folding. So again, we start with the basic DNA molecule shown here, which a single strand of DNA is only 2 nanometers thick. So very, very thin. Remember, a nanometer is 10 to the minus 9 meters. So 1 billionth of a meter long. Then the DNA, as you can see, is folded up around the histone proteins to form nucleosomes. So at this point, what we're talking about is a structure called chromatin. So this is the DNA wrapped around the histones to form nucleosomes. At this point, this initial wrapping then creates a thickness of about 11 nanometers. These nucleosomes are then folded into 30 nanometer folds of nucleosomes, so it's getting thicker. And then these highly folded nucleosomes are arranged in loops here. So we've now thickened up the original DNA to 300 nanometers, so we've packed it into a space of 300 nanometers. And these loops are then folded again into a section of a chromosome, which is now about 700 nanometers thick, and of course the double-stranded chromosome together about 1400 nanometers. So we've wrapped an awful lot of DNA, which is only 2 nanometers thick, into a chromosome that's 1400 nanometers thick. So we've packed an awful lot of DNA into this structure. Let's have a look at one more diagram and see how this relates again. This diagram is very similar to the two you've just seen, so it might be worth pausing the tutorial for a moment and just taking a look to see if you can follow the way that the DNA is folded up into the chromosome yourself. But you're looking at exactly the same thing as you've seen already. So you have your DNA molecule here in its double helix. It then gets wrapped around these proteins called histones. So you can see that shown here to form nucleosomes. So this is the nucleosomes. The nucleosomes are then highly folded, shown here. These highly folded nucleosomes are then looped, so these are called looped domains, to form a chromatin fibre, and it's these chromatin fibres that are then packed into a pair of chromatids, shown here, which makes up a chromosome.